let's talk cell cycle. One of the nicest things about cell cycle is that it's not a whole lot different in AP than it was in baby bio. Okay, so there's not really a whole lot of new information to learn. Really, this is about jogging your memory about what you learned back in baby bio. Before we get into the cell cycle itself, though, we have to talk chromosome. We have to talk about what chromosomes are and what it means to be a chromosome. So if I say the word chromosome to you, I think you probably have a picture in your mind, right? And that picture most likely looks like this right here, right? This little X-shaped structure that you're seeing circled on the screen there, right? Most of us think of chromosomes like that. And that comes from us learning about karyotyping and looking at chromosomes as they appear in a karyotype. But you're looking at chromosomes only during a very, very small window of opportunity during the cell cycle when you look at a karyotype. Most of the time, DNA isn't even in the form of chromosomes. And most of the time, the DNA doesn't exist in that chromosome two-arm shape that we're used to. Remember, when you see this chromosome, this is the original arm of the chromosome or the sister chromatid, right? But remember, this is a copy of the original piece. In other words, chromosomes don't get that X shape until after they've gone through replication, right? So that's why you're seeing here, this is copy of the same chromosome condensed. All right, so that's the thing. That's a replicated chromosome. Before chromosomes replicate, they would look like what you see here at the top. That would be what an unreplicated chromosome would look like. And then this is the replicated chromosome that you see here. And all we've done here is taken this X-shaped structure that we're used to seeing and just drawn those two arms of the chromosomes as lines, right? So a one-armed chromosome would just be that original piece of the chromosome. And then the two-armed chromosome is after replication has occurred. Now, why chromosomes? Not why chromosomes, but why do chromosomes exist in cellular replication or cellular duplication? It's because the DNA most of the time looks like that. That's chromatin, okay? And chromatin is all uncoily and messy and tangly. And in order to divide into two cells, we need to be able to separate out the original piece of DNA from its copy, right? We need to be able to sort that DNA out into different cells so that you're certain you have identical copies of the DNA in the two daughter cells that you make. Remember, this is a cloning process, so we want to have identical copies of DNA. So what we have to do is we have to take this chromatin and we have to do something called condensing, okay? And essentially we take this messy, all chromatin like that, and we coil it up into this chromosome structure. And by doing that, and actually we coil it and then we coil it some more and then we coil those coils, and that's what we call supercoiling, which we'll get into in another unit. But that's where you get that chromosome structure. When the chromosomes are all nicely coiled like that, it's real easy to sort the original piece of DNA from its copy. So that's why chromosomes even exist in the first place. But again, because they're for the purpose of moving the DNA, we don't actually see chromosomes all the time. We only see chromosomes during cell division when we need to move the DNA around. Otherwise, the DNA needs to be uncoiled in that chromatin form because that's how DNA is open and available for use. All right, so from baby bio, I hope you remember that there are actually three parts to the cell cycle. The first part we call interphase. The second part is the M phase or mitosis. And then the third part is cytokinesis, right? So interphase, the cell is just doing what it does. If it's a liver cell, it's doing liver cell stuff. If it's a skin cell, it's doing skin cell stuff, okay? Most of a cell's life should be spent in interphase doing its job. There are three parts to interphase. And throughout interphase, while the cell is doing its job, it's growing, right? It's maturing. And the first part of interphase is mainly a growth phase. It's called the G1 or gap 1 phase. Why gap? I don't know. I think of the G as growth, but it's technically the gap 1 phase. Now, in interphase, remember, we're not actively dividing, right? So the DNA actually doesn't look like chromosomes at this point. It would actually be chromatin. But if you saw chromosomes at this point, they would be this lowercase L shape or unreplicated chromosomes. We haven't replicated DNA yet. Remember also that the cell has a complete set of DNA. This is what we call diploid, right? Which means you have half of the DNA from the mom, half the DNA from the dad, and that means that you have these paired chromosomes. That's why you have the 2M. Okay, so you have a full, complete set of chromosomes, but they're not chromosomes right now. But if they were, they would look like that lowercase L. They'd just be a one-armed chromosome. During the S phase, which is the second part of the cell cycle, DNA gets copied. And in this case, then once you copy the DNA, that would be the first time you would be able to call to recognize a two-armed chromosome if the chromosome existed at that point. Now, the other thing that's interesting here is we've replicated our DNA, but we're still saying that the cell is diploid. Why? We have twice the amount of DNA in the cell. Why are we saying that this is a diploid cell? So think about human cells for a moment, right? You have 23 pairs of chromosomes. That's the diploid number, 46, right? If I take 46 one-armed chromosomes and I replicate them, now I have 46 two-armed chromosomes. Because I'm attaching the copy to the original via the centromere, I still have 46 structures. Okay, so a way to think about counting chromosomes is to think about counting the centromeres themselves. Okay, so think about you're counting the centromeres, not the arms on the chromosome. Even though you do have twice the DNA, you still only have 46 structures at that point if you're in a human cell. Okay, I hope that makes sense. There's an Amoeba Sisters video where they talk about counting chromosomes if you want to look that up on YouTube. All right, so once we've replicated our DNA, we go into the G2, which is also gap 2. It's just more growth, more preparation for mitosis. And then remember, mitosis has the phases that we abbreviate PMAT, right? Prophase, metaphase, anaphase, telophase. And remember, mitosis we often think of as being synonymous or with the cell cycle, right? We say cell cycle and mitosis as if they're interchangeable terms. But remember, the cell cycle is about the entire life cycle of the cell. Mitosis is really only about the nuclear division of the cell. All we're doing in mitosis is dividing the nucleus into two nuclei, and in the process, sorting out the DNA. And again, when I say sorting the DNA, that should clue you into the fact that we're going to need chromosomes here, right? We need chromosomes at this point. So the first time chromosomes actually appear are here. So even though I've been talking about chromosomes up here, you really have no chromosomes. You only have chromosomes starting at prophase. Prophase is the first time they appear. Some textbooks will put a step in between the prophase and metaphase and call it prometaphase. Some textbooks will simply call it early prophase and late prophase. It doesn't matter. Remember that these are processes that flow into one another. It's not prophase. Okay, now metaphase, right? The chromosomes move. It's a more fluid process than that. And so sometimes there are some distinctions made between the early parts of these phases and the late parts of the phases. Just don't miss the forest for the trees here. Okay, so we get prophase, metaphase, anaphase, and telophase, and we're dividing only the nucleus and the DNA at this point. And then finally, you have cytokinesis, and cytokinesis is the cellular division. So this is where you're actually dividing a cell into two different daughter cells. Okay, now there are some differences here between plant and animal cells. In animal cells, the cells kind of pinch. You get something called a cleavage furrow that appears between the two daughter cells as they kind of pinch off like a drawstring purse or backpack. And then you get two separate animal cells. In plant cells, that doesn't happen because the plant cell needs to lay down a new cell wall between the two daughter cells. So instead, you get a structure known as the cell plate that starts to get laid down between the two daughter nuclei, and that will eventually form the new cell wall. But either way, you're still dividing the cells there. All right, so we talked cellular DNA. This is what, if we made kind of a cycle diagram, this is what the cell cycle would look like. And notice the steps here are kind of proportional to how big they're drawn, right? This whole thing is interphase right there, from there to there, right? And then this is mitosis here, and then right here at the end, that's cytokinesis. Right there. Cytokinesis would be a real short moment at the end of the M phase. And then those two daughter cells enter the G1 right after that. The other thing you do need to know is that some cells do what we call G0. So if you have a nerve cell, once a stem cell that produces nerve cells creates a differentiated cell or creates a cell that will differentiate, that differentiated cell enters, instead of the G1, it enters a phase called G0. Which simply means that that cell is just going to grow to its normal size, perform its function, and die, and it's never going to go through mitosis again. It's never going to synthesize its DNA again, go through a second gap. And you may have heard that, nerve cells don't reproduce. That's all. All right, so let's look at what's happening in each step. Okay, so interphase, the cell's doing its normal job, right? After the S phase, you would have chromatids, sister chromatids, in other words, two-armed chromosomes. But really, this picture is calling these chromosomes, but they're not really chromosomes. That's chromatin at this point. You also have area called the centrosome, and the centrosome creates centrioles, but centrioles are only present in animal cells. Okay, plant cells would have other proteins that are present, but you're not going to see centrioles forming in plant cells. All right, so at the end of interphase, mitosis occurs, right? And again, my diagrams show a pro-metaphase, right? So at prophase, you see the chromosomes appearing for the first time, and that comes from condensation, in other words, coiling. Notice that the centrosomes start to produce these spindle fibers, right? And notice that the centrosomes are starting to move apart. So the centrioles are moving to opposite ends of the cell. And this process continues into prometaphase. Again, the nuclear envelope breaks down, and you're starting to see spindle fibers that are stretching between these two centrosomes that have migrated to opposite poles of the cell. And they attach to the chromosomes at something called a kinetochore. And a kinetochore is simply an area on the chromosome that serves as an attachment point for the spindle fiber. And that kinetochore actually can be responsible for some regulation of the cell cycle. And we're going to talk about that in a minute. All right, metaphase, meta means middle. It's appropriate. That's where the chromosomes are, right? They're in the middle of the cell or on the cell equator. At this point, every chromosome should be attached to a spindle fiber. And now what we've got is a game of tug of war, right? You've got this centrosome and this centrosome attached to spindle fibers, and they're each ready to tug on those spindle fibers and actually shorten them and pull those chromosomes apart. Literally pull this sister chromatid away from this sister chromatid. And that's exactly what they do. At anaphase, the centrioles or centrosomes will start to shorten those spindle fibers, pulling the chromosomes apart. And again, it's correct to now call this a chromosome and this a chromosome. They were part of the same structure, right? But remember, this up here was the original set of DNA. These were the copies that it was attached to, and now you're pulling the originals and the copies away. So now what we've got is a complete set of chromosomes here, a complete set of chromosomes here. And what you have is telophase, and telophase is where the nuclear envelope reforms, the spindles disappear. And by the way, plant cells do have spindle fibers, just so you know. It's just they're not produced by centrioles. And this DNA would start to uncoil and become chromatin again. Now, often at the same time as telophase is happening, cytokinesis begins. So here's our cleavage furrow, and you can see that what's happening here is you're starting to see the pinching inward there. And as telophase proceeds, cytokinesis is happening simultaneously, where the membrane is actually pinching into two daughter cells. Right? And again, if this were a plant cell, you'd have a cell plate. What I want you to notice about these pictures, if we go back, to the beginning, if you count the chromosomes, we have at the beginning here of mitosis, we have 1, 2, 3, 4 chromosomes, right? There are 4 chromosomes in this cell. Its diploid number is 4. If you get down to the end of this and you look, you've got 1, 2, 3, 4 chromosomes in this cell. 1, 2, 3, 4 chromosomes in this cell. So in other words, we started off diploid and we end with 2 daughter cells that are diploid here. Okay, so it's important to note the chromosome number here. Started diploid, even though we replicated the DNA, we weren't double diploid because remember, the copy was attached in the same structure as the original piece of DNA. So you start with 46 chromosomes, you keep that number 46 chromosomes throughout, you end up with 2 daughter cells with 46 chromosomes, but that's by pulling apart those structures, those chromosome structures. In baby bio, you heard about binary fission. This is prokaryotic cell division, which is way simpler than what you saw in mitosis, even though mitosis is not all that complicated. Instead of centrioles, the chromosome gets attached to the side of the plasma membrane. And what happens is the bacterial cell just starts to elongate. It as the original piece of DNA, which is attached here, and its copy is attached here, as the cell elongates, they get pulled apart. And when the growth is complete and the DNA is completely separated, then cytokinesis occurs. Real simple. There it is. Okay, so here's this mass, this clump of DNA here. There's an attachment point here and an attachment point here. Notice this piece of DNA remains attached to the wall here, and just as the cell elongates, those pieces of DNA get separated, and then you get cytokinesis occurring. Very similar cytokinesis to how you see in animal cells. All right, so what's new from baby bio is how is this controlled? In baby bio, we might have mentioned that there are these points in the cell cycle that are called checkpoints, right? These are points in time, spatial. They're not spatial points, they're temporal points, right? One important checkpoint, possibly the most important, is what we call the G1 checkpoint, right? There's a G2 checkpoint, there's an M checkpoint, there's all these different points where the cell kind of pauses, takes stock in what's going on, make sure that everything is proceeding normally, that everything is ready to go before moving to the next step, okay? As you can imagine, this is chemical signaling. It's just within the cell. Okay, so the way that the cell monitors progress is through monitoring chemicals. Oftentimes, and by the way, these chemical signals are referred to as a cell cycle clock. Okay, many of them are dependent on what we call CDKs or cyclin-dependent kinases. And these are protein kinases that are, as you see, ACEs, right, enzymes. They need a molecule called cyclin to be active, okay? So the MPF, the maturation promoting factor, happens when the CDKs that accumulate during the G2, the cyclin molecule. Cyclin is a molecule that attaches to that CDK. It accumulates during the G2 checkpoint, and once you get enough activated CDKs, that tells the cell, okay, we're ready to go. Then when you get past that G2 checkpoint and initiate mitosis, other enzymes start to break down the cyclin, decreasing the amount of the MPF in the cell. And then what happens is you start to see drops in that levels. And you can get into the M phase of mitosis more easily and allow that to proceed. So during that M checkpoint, if you have too much MPF, the cell's going to pause at that M checkpoint and wait until the MPF levels drop. So it's really monitoring levels of these certain chemicals in the cell. And if there's enough, you proceed. If there's too many, we wait until the levels drop, and so on. All right, so here's what that looks like, okay? So this line is showing you cyclin, right? Okay. CDK is present in the same amount each time. But as this cyclin increases, right, you start to attach that to CDK to produce the MPF, right? That amount of MPF peaks at the M checkpoint right here, okay? So it's going to peak right there. And then at that point, other enzymes are going to break down cyclin. So there's the cyclin component, there's the CDK. And once that cyclin gets broken down, you deactivate CDK. And so you're going to see while the cyclin decreases, the CDK is still there. It's just in an inactive form. And then once you hit the next G1, you're going to see movement up of the cyclin level. So you get these cyclin levels that go through these cycles themselves along with the cell cycle. All right, so there are some other internal and external cues that determine how fast or how long it takes for mitosis to happen. The first are internal cues. At the M phase checkpoint, it actually occurs at metaphase, and that's a point where these kinetochores that I mentioned become really important. Kinetochores are capable of signaling the cell if they're not attached to spindles. So if the kinetochore is not occupied by a spindle fiber, it sends out chemical signals that will halt the cell from going any further until every spindle has attached to every chromosome. That way you don't get chromosomes that fail to separate. Like if right here, this one is not attached to a spindle, they would both get pulled in the same direction. But if each side is attached to a spindle, then the spindle is going to pull it apart like it should. So those kinetochores are really important for that reason. And then there are also external cues, hormones in a multicellular organism from other body cells can promote growth, stop growth, and so on. And then you have something called density-dependent inhibition. We call this contact inhibition in baby bio, where cells are touching other cells on all sides. And if they are, and they're all packed in nice and tight, then they really don't have room to grow and divide. When you interrupt the contact inhibition, when cells die off, then that triggers neighboring cells to grow and divide to fill in any gaps that the dead cells leave.